In this picture, Watson and Crick are shown admiring this model of DNA, and the model of DNA was exciting to them partly because it immediately suggested a mechanism for replicating the DNA, and that mechanism is those hydrogen bonds between the bases in the nucleotide base pairs that can get unzipped easily. And so that's what Chapter 16 focuses in on, and we're going to see how these get unzipped and separated. And so there'll be some images of this flat, untwisted ladder, and we'll talk about the legs and rungs of the ladder some. And so this mini lecture is going to be about DNA replication, and here's the first image of it. So there's the DNA double strand, it gets unzipped, and then it gets replicated. And this process of replication is called semi-conservative, and it's to distinguish between the different possibilities. One possibility is that you would have a parent molecule like this that's blue, and after the DNA was replicated and the cells divided, maybe one cell would get the blue DNA and the other cell would get the newly replicated, in this model, light blue DNA. That's called conservative replication. And this is semi-conservative. Each daughter cell receives one old copy of the DNA and one new copy of the DNA. And then the next thing is see that's using that same light blue, dark blue motif in this figure that's just from the book to show two separate DNA molecules getting replicated from one parent. And this is the same model that would be used for DNA replication in any organism on our planet. And then the next thing to mention is that the DNA is going to get unzipped and replicated. But DNA strands are really long. So in one of our chromosomes, there might be 100 million letters. And the decopying process only goes about 1,000 nucleotides a minute. And so it takes a while to copy 100 million. So instead of having it unzip and copy from one end, this is what happens, is that at many, many sites along the length of the chromosome gets unzipped, kind of like a zipper breaking in the middle, like that, starting to get separated. So that's what's being depicted here. And each of these sites is called an origin of replication, and the DNA sequence at that site identifies it as such. So when it's time to start S phase, to go from G1 to S phase at the start of the cell cycle, the so-called G1 to S transition that we learned about back in chapter 10, I think. And at that point, there are proteins that bind to the origin of replications and start to unzip them. And then the enzyme that does this work in replicating the DNA is called DNA polymerase. I've got to draw it over here in a different color. We've learned some about RNA polymerase. And RNA polymerase and DNA polymerase are similar in that they both use DNA as a template, but then they're very different. And it's really important for students to distinguish between the role of RNA polymerase, which is involved in gene expression, and RNA polymerase just copies portions of the DNA in the genome. The RNA polymerase copies the transcription unit of individual genes. So along the length of this DNA, the RNA polymerase would just copy short patches in the process of gene expression. And that's very different than the role of DNA polymerase that's going to copy the entire chromosome for a very different purpose. This is during DNA replication. The chromosome will be replicated and separated. So we want to distinguish between those two. Both of them unzip the DNA and use the DNA as a template, but for very different functions. Okay, so then if you look at this, there would be some DNA polymerase here, and here, and here, and here, and here, and here. So we have lots of molecules of DNA polymerase doing this work, copying the DNA. And we're going to see that the book zooms in on this, and this region, here it is circled here. So that region is called the replication fork, and these unzipped areas are called replication bubbles. And so in one bubble there are two replication forks, and the replication forks are all comparable. So there are many replication bubbles along the length of a really long DNA chromosome from a human, but the story is all happening at the replication forks, and all the replication forks are the same. And here's the first thing we want to learn about the replication fork, and that is that because the DNA is anti-parallel, and DNA polymerase can only go one direction, we have this situation where this would be 5' to 3', that direction, and it's copying this anti-parallel strand, so the parental template would be, this template here would be 5' to 3', and so then the lower template strand would be 5' to 3' here, and so consequently, because they're anti-parallel, this DNA polymerase needs to head that way. So we have two strands being copied at each replication fork, and at every replication fork, one of the strands is relatively easy to copy because it's DNA polymerase heading in the same direction that the DNA is getting unzipped. The other strand is harder to copy because DNA polymerase frequently needs to start over because it's going in the opposite direction as this unzippering effect. Consequently, there's the easy to replicate strand that's referred to as the leading strand, and the slower replicating strand is the lagging strand. And you can see that effect here, where on the leading strand, there's just one solid blue, because one DNA polymerase could grab and hang on and keep going. DNA polymerase is said to be a processive enzyme, and a processive enzyme holds on to the substrate and catalyzes the same reaction over and over before letting go. So DNA polymerase holds onto the DNA template and then just goes sliding. One DNA polymerase slides down it and copies thousands of nucleotides before it randomly lets go. And so in this at this replication, there's a leading strand heading that way, and that DNA polymerase can keep copying it like that. Okay, so now I'm going to zoom in to the replication fork like that. We've just zoomed in here. You see on the lagging strand, it's a fairly simple story to tell. The DNA polymerase got started, and here's DNA polymerase, and it's just heading down. Now, the book introduces several proteins and describes them that are helpers. The bottom line is DNA polymerase is getting its job done, but here's some helpers. Here's a protein that helps unwind. It's called a helicase. It helps unwind the DNA because the DNA is in this twisted anti-parallel structure, and the helicase is going to help untwist it. Then here's the next thing is there's an enzyme called primase that's helping DNA polymerase get started. That's covered up here. There was a primase up here that got the leading strand started, but we only need to prime the leading strand rarely. On the other hand, the lagging strand has to be primed repeatedly to help DNA polymerase get started because the DNA polymerase has to go that opposite direction. And so let's just tell this story down here on the lagging strand a little bit because it shows a little more of the details of the replication process. And this primase is going to make a product, this red product is actually RNA, and the RNA is eventually going to get eliminated, and so that the strand will be DNA. But initially, there's a short product that's called a primer. So primase makes an RNA primer that lets DNA polymerase get going, and then DNA polymerase takes off and does its job. Then here it just shows that DNA polymerase needs to help eliminate the primer. We'll talk about that is left over. And then a final enzyme will help glue together any gaps. There are also DNA repair enzymes, and so if DNA polymerase makes mistakes, it can be repaired. So we want to zoom in on this DNA polymerase enzyme and ask, what does DNA polymerase need to do to get its job done? And DNA polymerase needs the following things to get its job done of copying the DNA. The DNA polymerase needs a template. Then it needs a primer. And the primer is like this. It shows it DNA polymerase where to get started. And then the DNA polymerase also needs the nucleotides, the letters that it's going to use to join together to make the new strand. And so the nucleotide letters are nucleotide triphosphates. So like DATP and DGTP, DCTP, and DTTP. DATP is very similar to the ATP that we learned a lot about in our chapters on metabolism. The only difference is in the sugar, this DATP is missing an OH group that the ATP has. But remembering that this is a high energy intermediate, you'll be able to easily understand that these guys get added by DNA polymerase and also provide the source of energy needed to synthesize this strand. And this synthesizing this strand is an exergonic reaction, energy requiring reaction. And so it's just important to appreciate that that energy is held in these nucleotide triphosphates. And when ATP is added, when DATP is added, two of the phosphates are removed. And so if we just scroll back to this image for a second, see this is, for example, DATP with two of the phosphates removed. And the energy used from those drives this synthesis reaction. So that's what DNA polymerase needs to get its job done. And then these replication forks will keep going until they reach the other bubble. So this one will keep going this way until it reaches another replication bubble, and when it does, the synthesis will be complete, like that. And so this is happening in vivo. That means inside a cell. And those are the highlights, and what we want to do next is we want to take what we're learning in Chapter 16 about the natural DNA replication, and then in these figures in Chapter 20 that we're going to focus on next, that DNA polymerase is used in some of these methods. DNA polymerase is used in a test tube, so in a purified test tube, to do DNA replication, that copying, in a test tube. So scientists will purify the DNA polymerase and the other components, and they'll add the necessary ingredients to a test tube, and they'll get that DNA synthesis process to happen in the test tube. And that's next.